welcome everybody. Thank you for joining this. I'll just explain a little bit very quickly about what we're doing and why. And then this is really for the nodes to talk about their activity and to sort of share their thoughts on progress and how everything's going. So as everyone knows, the platform was set up to be the combination of a number of different nodes, beginning to think about how they work together and build on each other's expertise. And we're involved with my colleagues here in running the hub to help facilitate that. So it is a platform. It is a combination of the 11 nodes, research programs, some in methodology and cross-cutting, some in particular disease areas. So a wide diversity and a desire to make the whole worth more than the sum of the parts. The sum of parts is really impressive, but the idea is that you want to make it even more than that. So as part of this, we have the nodes, we have our management group, but we also have a program advisory board. And in our program advisory board, there was a real, with people coming from all manner of different backgrounds, be they research, industry, patients, funders, and there was a real desire from that to find out more about actually what's happening on the ground with the nodes. And this seemed like an excellent idea. And therefore, this meeting came from a combination of those meetings and a desire for everybody to find out how everybody is getting on, what are the challenges and the successes, and what are the areas of research. So this is the first of two showcase sessions that we've pulled together to cover the 11 nodes, and so people can hear about it. So it's as simple as that. This is about hearing about research, hearing about opportunities and progress, and learning how we can all work better together. Now, those people in the nodes will be aware that we are now going into a reporting cycle to MRC, who are the coordinating funder. And so this will hopefully help with people as they're preparing their paperwork for the reporting about progress. So that's what we wanted to do, make this a really interesting, dynamic session about how it's all going and the science. Now, we are quite tight for time. So if people could just keep half an eye to that. So we do need to cover the 5 nodes that are speaking today. So what I will do at that point, unless any of my colleagues from the hub want to add anything, I suggest that we just crack on. Now, Nikita, how do you think we should run questions? Should we have people putting their hand up? It's always difficult with 5 pages of people. Have we thought about how we should run questions? Either put chat, the Q&A function, or raising hands. Have we thought about that?
We do have a Q&A function on top, so anybody with questions, they can just type in their questions there.
Yeah, so type your questions in, then we'll come to the speakers in the Q&A session afterwards. Okay, lovely. And please do, half the benefit of these types of sessions comes from the discussion. So at that point, I shall pass on to Sid Banker, who's presenting on behalf of EpiGenRare. Sid.
Thank you, Dave. Could I have my slides, please? Thank you. Great. So, yes, thank you very much for the opportunity to discuss the work happening in the EpiGenRare node, which stands for Epigenomics of Rare Disorders. And thank you. So next slide, please. Great. So the EpiGenRare node is coordinating research in the domain of epigenetic diseases, which primarily we define as chromatinopathies and imprinting disorders. Chromatinopathies are conditions which are caused by changes, variants in genes that encode for different components of chromatin control. So if you look at the human genome, there are about 900 to 1,000 chromatin genes, out of which almost 100 are now associated with a human disorder. And imprinting disorders are those which are caused by defects in imprinting. So overall, if you add the number of patients who are affected by epigenetic diseases, the conservative estimate would be somewhere between 1 in 100 to 1 in 500, and perhaps it's a bit more frequent than that as well. But we also proposed that we would like to function as the hub for epigenomic expertise for other rare diseases as well in the network. Next slide, please. So the research challenges that EpiGenRare is addressing are threefold. And on the right side of your screen, you can see a diagram which illustrates the three main projects that we started the node with. The first one is around deciphering the genome-epigenome relationships. Now, I don't really know who else from EpiGenRare node is present at that meeting. A bit like what you said, David, there's so many people on the call. So if there are other EpiGenRare node members, and if I describe their projects incorrectly, please do correct me. But the first project is primarily being led by Professor Eamon Maher, who was at University of Cambridge and is also at University of Ashton now, and Deborah McKay from Southampton. So here we are attempting to create a linked resource of patient samples to decipher the genome and epigenome relationships in imprinting disorders. So imprinting disorders, for a large number, for a subset of patients, their underlying reason why those imprinting disorders occur in some patients is poorly understood. There's some evidence to suggest, at least in some patients, that there are genomic changes which result in these epigenomic alterations. So we're really trying to create this linked resource to be able to decipher those unknown relationships. Second, because there are such a large number of chromatinopathies, and many of these chromatinopathies have overlapping biological and molecular pathways. So we are attempting to explore the possibility of therapeutic convergence, which means that could we have same therapeutic approaches or molecules to treat multiple disorders that have a biological overlapping, molecular pathway overlapping. And this project is being led by Albert Besson. I don't have the pointer in my control, but Albert's photograph is, I was pointing through my mouse, but I just realized that probably no one can see my mouse. So Albert's photograph is the first one, and Eamon and Deborah's photographs are next to Albert's photographs. So Albert is he's an expert in mouse modeling, and he's also one of the PIs for this EpiGenRare project, and he's created a number of mouse models, and we're currently analyzing their phenotypes. And we've got a relationship, an agreement with a pharmaceutical company to supply us drugs to test the therapeutic convergence hypothesis in these mice as well. And the third project is by using human stem cell models in chromatinopathies, and that project is being led by Deepak Srivastav, Christina Diaz, and Sue Kimber in King's College and University of Manchester. And here the idea is to lay ground for large-scale, high-throughput drug screening for therapies using human stem cell models. And we're creating a number of cell models, which we will also be very happy to share with anyone else who would like to study these diseases. And as I mentioned at the beginning, in addition to these three projects, we will also provide epigenomic expertise, access to state-of-art platforms, pipelines for epigenomic analysis, and clinical expertise for epigenetic diseases to other nodes of the platform. Next slide, please. So we were told to divide this presentation under these headings. So in addition to the research projects, the other very important aspect for EpiGenRare is networking. So although the EpiGenRare node application was led by 5 individuals, we're very keen to expand this and create an international network of researchers, clinicians, industry partners, and patient support organizations who are interested in this particular area. We already have agreement from 60 members, and this network is actually growing quite rapidly. We've organized an introductory scientific meeting for EpiGenRare node for 30th of October later this year. From our network, we've also invited 5 individuals to be part of EpiGenRare advisory board. That includes fundamental science researchers, clinicians, patient organization representatives as well. We're very keen to network with other nodes, and sorry, this need not be repeated from the previous slide, but I would like to highlight that we're organizing an EpiGenRare conference on 26th of March in Manchester, and this will follow the UKRD conference on 25th of March, which will also be held in Manchester. So if there are other node members who want to learn more about what's happening in the node and learn more about the area of epigenomics in rare diseases, I'm sure that you'll find this conference very interesting scientifically and clinically and from a networking point of view. Next slide, please. Then we were told that one heading needs to be around achievements and activities. So our node actually was delayed to start because of organizational reasons. So we're coming to about a year of our node, and so far we've had from the node 3 publications, and there has been a grant submitted as well. We've actually also done some interesting PPIE work, which I think will be of relevance to a few other nodes as well. So we've actually been developing a new AI large language model-based model for co-developing patient information resources for epigenetic disorders. But actually the model would work for any disorder. So if there are groups who are interested in using this model for developing patient information resources, they're very welcome to contact us and collaborate on that. We've also included patient partners in the network and the advisory board, as I mentioned previously. We'd also promised to organize family education days, and we held a meeting for Kabuki syndrome, which is a chromatinopathy and an epigenetic disease. We held a meeting for patient support groups, and about 75 families from UK, but also a few families from abroad attended this meeting earlier this year. Next slide, please. So I think this is probably the last slide, challenges and impact. We think, as I mentioned, there are 900 chromatin genes, only about 100 or so are so far associated with human disorders. So we think that there is opportunity to discover new disorders, and also with the linking of epigenomic and genetic data, we think that we will be able to identify some new imprinting disorders as well. We want to improve diagnosis, so we're doing a lot of work around using DNA methylation-based episignatures for improving diagnosis of these disorders. Through our stem cell and mouse modeling, we're hoping to improve mechanistic understanding and discover treatments. There's also lots of opportunities coming up for clinical trials in these disorders, and we're already in talk with a few pharmaceutical companies or trial sponsors, but some academic trials as well, potentially in this particular space. And we're hoping to be able to collaborate with the trials node in UKRD, but with other experts as well in this platform in conducting these clinical trials. We're really very keen to collaborate with other nodes. We're wanting to involve patients and support groups and make knowledge accessible through our efforts around AI and large language model-based approaches. I think that's my last slide. We can just check. Yeah, and lots of people to thank, mainly Albert, Eamon, Deepak, Sue, Christina, Deborah. These are the people who are leading the projects, but we've had really great input from Jason Lurch, Andy Sharix, Dan Whitaker, who in terms of organizing these nodes, and Sarah and Brendis in terms of they are the PTRAs who are working on these projects, and Henry and Gabrielle, who are the project managers for us. I've put our Twitter and LinkedIn handles, and there are QR codes if you want to scan and follow the activities of EpiGenRare node on these social media platforms. And just at the end, I would like to highlight the EpiGenRare conference on 26th of March 2024. We're hoping to release more details about the conference next month. Thank you very much for your attention.
Thank you very much. Very clear and very interesting. And can I suggest we just leave that slide up about the details for the conference and how to contact. And the next is the CAPTIVATE node, who are doing this, I understand, as a team. So it's a sort of tag team presentation. So over to CAPTIVATE.
Thanks, Dave. So I'm going to kick off. I'm Cindy Billingham, and I'm the director of the CAPTIVATE node. And yeah, we're going to try and seamlessly run from one of our team members to another, but you will see pictures of them in a moment. Next slide, please. So this is a busy slide, but if I can just introduce the CAPTIVATE node and remind everybody what we're here to do. We put ourselves, you'll see in the middle of the slide here, we put our node in the center of all the other nodes as part of the whole hub, because I think we will be feeding in and feeding back from many of the other nodes to try and support them in their clinical trial activity. And I think you'll see in the challenges, that's definitely one of the sort of things that we still need to really try and understand. So these showcases are really useful for that. And then on the left-hand side is the idea that we have to get input from investigators, project partners, specialist expertise. So, working with the clinicians, other trialists, patient and public partners, industry and regulators, to understand their positions on various things and their current knowledge and their needs of the CAPTIVATE node. And then on the right-hand side here, the outputs that we will then generate, which will be supporting, going forwards, supporting the community in being able to do trials, hopefully more easily, and therefore directly affecting patient benefit. And just at the middle at the top here, the 3 key research projects we're looking at that we'll talk a little bit about in the next couple of minutes is that one idea that you can't do multiple phases. So trying to do adaptive designs and looking into how we do adaptive designs where you can try and do just a single study to then get treatments approved for rare diseases. The second sort of key project is, the key thing here is about maximizing external information, and that will get us away from potentially having to use randomized designs. So we'll become more efficient in that way, but you need good use of external data to then lower your sample size numbers or make the most of the patients you have. And then investigating how low you can really go. And a lot of Alex's work, and he'll talk in a moment about this, but a lot of Alex's work has been about how we make the most of small numbers of patients in a statistical way. And as I say, he'll talk about that. Next slide, please. So this is the team. On the left-hand side, you'll see all of us talking today. Myself and Tori, and Alex is paid on the grant, so he's the real worker here. And Tom is our project manager and has really revolutionized our activity since he arrived in March. And then the top 2 on the right-hand column, which is Stephen Julius and Matt Burns. So Matt is also paid on the grant, half of his time, and they're based at Sheffield. And their key role has been in the what we call N of 1 trials, this really, really small numbers, this extreme rare diseases, and the statistical issues and how we run trials in those very small numbers. Carol Gamble from Liverpool is key to a lot of our PPIE work. And then it's worth just pointing out Tim Barrett and Pam Cairns bring very much clinical expertise into our node, as well as obviously linking with others. And just to highlight, Simon Gates here is the lead for the LifeArc part of the essentially the team that are going to actually run trials in rare diseases along with other collaborators. But Simon leads on that, so he's a key link between the LifeArc funding that we have and the CAPTIVATE node working on the methodology. So the two will interact with each other very directly, which is really neat, hopefully. Next slide. So going to talk about progress, updates and upcoming activities. Next slide, please.
Okay, so yeah, as Cindy said, a lot of the focus of the work at the moment has been on trying to make the most of very, very limited data sets. To that end, I've been looking at just modifications to sequential designs for single-arm trials. And I've got a poster to take to the International Clinical Trials Methodology Conference at the end of this month that just goes through a way that you can kind of opportunistically make sequential designs a little bit more efficient in a binary endpoint case. Beyond that, there's some work I've done on something called confidence curves, which is kind of it's a way of capturing a lack of information in a robust way that you can calculate and make predictions of things with. Very similar to probability distributions, just in a different kind of space, and that forms the core of a lot of the work that I'm looking at. But in addition to the kind of the statistics side of things, there's kind of two other main arms we were looking at in terms of how you actually like set trials up and manage things like registries, but then also PPIE. And to that end, we are putting together a focus group meeting in October or November, I think. And we have a PPI paper that is going to feed into that, feeding the kind of the current state of research in PPI in rare diseases, bringing some of the, like condensing that into a form we can bring to the meeting and discuss with PPIE partners, and then capture the outputs of that to form a kind of resource that could be used in the future. In addition to that, there's some more kind of statistics theory papers that are currently in progress in terms of, calculating with these confidence curves, actually capturing the contents of this poster in a paper, and some other approaches to things like quantile regression in a statistically robust way. Next slide, please.
Okay, I think that takes me on to myself. Thank you, Alex. So just to elaborate on a couple of things that Alex had said there, CAPTIVATE, along with, I hope, a number of other members of this call will be at the ICTMC conference in Edinburgh at the end of the month. So please do reach out to us either after this call or ahead of that conference. If you are there, I'm sure members of the team would love to speak to everybody. In addition to that, the PPI focus group meeting that Alex mentioned will be hosted on the 19th of November. The idea behind this is we want to get patient and public feedback on kind of lived experiences within trials at this point. Also kind of asking key questions around what levels of risk that patients are happy to undertake when undergoing clinical trials. Also any attitudes and feedback they have on some ideas that Alex has had around new and innovative trial design, especially around placebo drugs and things like high toxicity in treatments on clinical trials. So that meeting will be held in November. We're hosting it in Birmingham. And obviously, Alex mentioned that a paper will come out following that will hopefully take some of the ideas and discussions from that meeting and release that to the public. Some of the things coming up in the next couple of months, as we look towards 2025, there's two real key focus groups that we want to get going. The first one is a methodology focus group. We want to invite other people that are already researching trial methodology and trial design into a room, get them in a room together with ourselves, not only just to share ideas and research focus, but also just to prevent duplication of efforts. We want to make sure that what we're doing as CAPTIVATE aligns with what's already out there in the academic space, and make sure that we're not duplicating effort with other groups. But also using this group as a somewhat of a peer review body, so that we can present and discuss ideas that we've already looked into over this past 12 to 18 months and get feedback on what we've been doing to this point. And also the overall aim of that methodology group would also be part of helping to build our library of academic resources. You know, attending that methodology group will bring together everybody's work and research to date, and hopefully we can start to flag, signpost, and bring those together to create a library of resources. I think following on from that methodology group, that try and launch that in the first quarter of 2025, and then following that we'll be looking at a regulators focus group, so looking really at what regulatory hurdles are coming, what exists now and what are coming in the rare disease clinical trial space. Also asking the questions around regulators and what compromises would they be willing to make and to consider when we're looking at trial design for rare disease research, particularly when we've got low populations, and also asking how we can better communicate the risks to patient groups within these trials so they understand fully what they're taking part in, essentially, and they can be fully informed. And then finally, one of the main questions we want to ask that group as well is, what is the barrier of evidence that we can get when it comes to licensing new drugs and new treatments coming out of clinical trials, especially when we've got such low data coming out of rare disease trials? So what is that barrier to entry for evidence that could be accepted? So they're kind of two main focus groups that we have coming up. Other ideas that we have, and I think it was really useful that it was mentioned during the EpiGen presentation around CAPTIVATE really being a node that can collaborate with every other node within the rare disease research network. So some things we want to possibly launch is something called office hours, or maybe a different name, but the idea being that either once a week, once every 2 weeks, we would have set hours and a bookable system where probably launching it initially to the other nodes could drop in and speak to members of the team, so Alex, Tori, or Cindy, depending on kind of the questions required or the research area that they want support with. And we can kind of start to collaborate on trial design ideas and methodology with other members of the nodes. And if this is successful and if uptake is good, then look to try and launch that outside of just the RDR network and to others within the rare disease research space. So watch out for that. More information will come forward on when office hours will launch. And kind of building on that finally would be, if this is all successful and we do get good feedback from kind of office hours, is then start to possibly launch a series of workshops around trial design. And that would really be more aimed outside of the rare disease research network and how we design trials and how other groups and areas design trials. But that's me. I think next slide, please. And again.
I'm talking about the impact when I've unmuted myself is what I'm going to say. So you've heard from various of the members of the group that we have a series of academic papers and conferences that we are planning on attending and publishing in. And obviously as academics, that kind of forms much of our bread and butter. But beyond that, and you've heard it touched on, we are planning this kind of library of resources that will include sort of training materials, generalizable trial protocols, and other study documentation, like sort of skeleton wording that might go in an analysis plan or in information sheets, for example. We are also planning on producing some guidance about the actual setting up of these trials in the rare diseases in the landscape when we're using these designs and the sample sizes are so low. Again, the idea being to help with where we've had those conversations around sort of with regulators around their barriers. We may as well share that so everyone can feed off it rather than everyone having to sort of repeat the task. For statisticians, of which I imagine that we're probably the only node represented with statisticians here, but we are planning on producing a series of sort of user-friendly statistical programs and packages that allow these designs to be implemented day to day and sort of wider than this group, because that will obviously be one of the biggest barriers for the implementation of these methods sort of externally. And we're also planning on making available in this library a series of case studies and examples of where these things have been used. And the whole idea is that when all of these are packaged up and will become available sort of publicly, that then gives the wider research community, both within the RDR network and wider, the toolkit to be able to design trials in these smaller populations. And I think the real key point here is that if these resources are available and people are aware of them, then the number of trials that are designed in these rare populations, they will increase, and there will be hopefully therefore the real step change in the actual treatment of patients with rare diseases. Next slide, please.
Thanks, team. This is our last slide. Just to very briefly mention the challenges. So time constraints and competing projects, Tori and I certainly, and others, multitask CAPTIVATE alongside others because we work in a busy trials unit. In some ways, that's an advantage because we have access to real world running of trials and understand rare diseases and trials already in rare diseases. But it's definitely a major focus for Tori and I and a high priority, but we do have to juggle alongside other things which can be challenging. And just this is really useful, the showcases, but making sure that we understand what the nodes need and other clinical groups. And for example, I'm going to the Oxford Harrington Rare Disease meeting next week so that I can get an understanding of that clinical and scientific need from that particular group. And I think it's just trying to engage with all the different groups is quite challenging when there's so many. Can we overcome the regulatory hurdles? We hope so, but we're not sure. But so that will be a challenge, I think, trying to get the regulators on side to compromise. Ever change, it's a constantly developing field. It's amazing how active this field is of rare disease trials. So just trying to keep on top of all that is quite challenging, but exciting as well. And just making sure that people know about us and what we're doing so that there isn't duplication and that we can sort of interweave with other methodology groups and make sure that we all work efficiently together. And that's it. Thank you very much. Oh, and just the final challenge, by the way, I did notice, Alex's work is quite technical statistically. And I just thought of, while we were talking, I think an extra challenge here is making sure that some of the statistical work that we do, which is quite technical, becomes very accessible and understandable for the wider community. And that's absolutely what we will endeavour to do. Next slide and final slide is just questions and answers, I think. That's it, Dave. Thank you.
Okay, wow, fantastic. There's so much to talk about there. We do have to move on. I've had my reminder message that we need to move on, so we'll move on. And the next is the ELSI node and Ramona. Ramona.
Yes, hi, and thanks very much for the opportunity to present an update on where we're at. Right. So I think we can go to the next slide, please. And I thought this is just an important, just a quick overview, I guess, on about our nodes and the projects you will hear a little bit more about in the next few minutes. But basically, our node, or the ELSI node, is looking at the ethical, legal, and social issues in rare conditions research and clinical practice. And our node includes 3 separate projects led by the University of Manchester, the University of Oxford, and Cardiff University. I don't think Annika or Angus are here with us today, so I'm going to try to give just a quick summary of their projects as well, but very happy to pass on questions if there are things a bit more detailed than I can discuss. Basically, what we're trying to do is to address a few issues we believe are essential, and I think quite a few have been mentioned already. So we're trying to understand in the first project the impact and outcomes in rare conditions, clinical trials. And to this point, we're trying to really, and I think I see Cindy smiling, so I think we're on the same page with quite a few things with the CAPTIVATE node, and I know a few others as well, but really trying to develop capacity for 2 things. One is a meaningful set of core outcome sets and patient-reported outcome measures. And I'm happy to discuss that in a minute. But really, with this, we're trying to capture the needs of patients and family members and make sure that these are expressed, captured in clinical trials, perhaps a little bit more than in our experience they tend to be reported or factored in at the moment. Annika and her team is leading a project on a patient's life with undiagnosed rare conditions. Some of you may be familiar with Annika's work already, but what she's trying to do in this project is really understand the experience of those living without a diagnosis, and to try to consider ways in which patients and families can be supported. And she's working with her team, of course, with patients, clinicians, to develop resources to support them. And Angus in Cardiff and his team will be looking at mainstreaming genome-based investigations across specialties. And some of the things he will be looking at will be some of the ethical, but also some of the communication and counseling aspects and barriers of incorporating these investigations into mainstreaming. And again, the hope, and you'll see that in a minute, is to develop some tools and some training opportunities to address some of the barriers we anticipate, or we will most likely be able to explore in more detail quite soon. I'm happy to give a very short overview of the research progress so far, which is not great. It's on the next slide. In the sense that we were a little bit delayed, I think, like other nodes as well, for various organizational reasons. And whilst we're waiting for ethics, which is something we're all doing, or recruiting more members to our teams, I think both our projects here in Manchester, but the others as well, are trying to use some of the secondary data we have access to provide a good background for the research or the projects we're planning to implement. In the next slides, you're going to see two publications that are either, so one of them is already published, one was currently accepted for publication. And what we're doing here, or what you will see in these papers, if you want to have a look, is our findings so far about where we are in terms of the patient-reported outcomes and core outcome sets. And I'll stop and just explain that for a little bit. So when we talk about outcomes, and by outcomes I mean, especially in the context of clinical trials, results of care. So that can be biomarkers as discussed, or various other outcomes reported either by the clinicians or the patients. So we look at all these outcomes, and especially we wanted to look at patient-reported outcomes. So these are reports coming from the patients. And one example could be quality of life, and this is something we know is a primary outcome of relevance for patients. It's probably fair to say it's the most common patient-reported outcome. So we ran an analysis and wanted to see in the current or in clinical trials for rare conditions at the moment, what are the main outcomes and patient-reported outcomes reported. And we were quite surprised to see that only about a third of the current trials include patient-reported outcomes. Like, there's, of course, there could be many reasons for that, and I know there'll be many comments and questions, but I think it's probably fair to say that some of the evidence we use at the moment when we talk about the success or the impact of a treatment or of a clinical trial doesn't often or always reflect patients' views and experience. And we're trying to understand, certainly in the first projects that you saw, is how we could best reflect patients' hopes, expectations, and needs in the clinical trials that are designed for them, but as I mentioned, don't often include their views and experience. So that was the focus of one publication, looking at patient-reported outcomes, and the other one captures the current core outcome sets used for rare conditions. So a core outcome set is a set of standardized variables that should be measured and reported in all clinical trials. And this is emerging research, I guess, and methodology, and quite essential if you think of all the challenges when working with rare conditions like small patient populations or limited data on disease progression, treatment effectiveness, lack of validated patient-reported outcomes, and so on. So having a standardized set of variables that should be measured or could be measured and reported in all clinical trials is quite essential. And we found that there are not too many out there. I think there are about 8 core outcome sets for rare conditions. So it's, again, we highlighted, I think it's probably fair to say, the need to improve this. And it's something we're trying to do a bit more in the project. We were very lucky to be able to secure some funding to advance these conversations. So we were funded by the Brochure Foundation to do a workshop around rare diseases, genomics and justice. So this will take place in January in Switzerland. We also did, on the next slide, again, whilst we're waiting for some of the organizational details and admin to be in place. We did a number of external talks and workshops at various conferences and collaborators where we highlighted some of the findings we have or some of the priorities we would like to focus on, again, with the aim of widening our network and trying to bring as many stakeholders into this conversation as possible. I think, like most nodes, we have a number of meetings and newsletters and conversations with our scientific advisory board, which includes clinicians and scientists, patients' representatives, bioethicists, and so on. So we're really trying to learn from everyone's background, personal and professional experience in everything that we do. The next slide includes a few other I guess activities we've done, some of them quite creative. So we've been involved in rare disease days, for instance, or family days. We've also tried to disseminate some of our work and to share some of the patient's experience living with rare conditions via podcasts and more artistic products as well, if I can say that, a short documentary film that we did. So on Rare Disease Day, we asked patients and families, clinicians as well, took part. You'd be surprised on Rare Disease Day, and they sent us clips of their life on February 29th this year. So we were able to put together a short documentary with a day of rare conditions. I'm very, very happy to share the link. It's free for everyone to have a look. So again, we're trying to mix and match some of the scientific, more patient-focused, more artistic-focused, public-oriented outputs and activities as much as we can. We also have a few upcoming activities on the next slide, so you're very, very welcome. I think Sid mentioned one of them or two, like the conference is coming in March, but also an ELSI Nodes Day in March 2025. I'm going to send a few links as well, but until then, there's a workshop focused on rare conditions taking place in Manchester on October the 1st. You may have seen some comms around that, but we're trying to spend a day talking about the main ethical, legal, social issues around rare conditions. And it tends to be, for those of you who know genetics, a very successful event, workshop. So we're very, very excited about that. And a few other, workshops and meetings, including Songs of Genomics in Oxford in a month or two. In terms of more scientific, I guess, objectives, so that's the next slide. Basically, once ethics is in place, and I think that's the case for everyone really, and by everyone I mean our team here in Manchester, but also the teams in Oxford and Cardiff. Once ethics is in, we're going to progress with data collection. As you saw, we're mainly doing secondary analysis, pardon me, at the moment. but we're planning to start collecting data from patients, parents, clinicians in all 3 sites before the end of the year or early 2025. I think our hope with this, and that's the next slides, although I'm conscious that we're asking big questions, I guess, is really to try to develop capacity and meaningful ways to support clinicians involved in recognizing or investigating or supporting patients with rare conditions. So that could mean core outcome sets, for instance, or patient-reported outcome measures to be used in clinical trials. That can mean resources for clinicians and patients with undiagnosed rare conditions, or various tools that can be used to better communicate and support incorporating genome-based investigations into mainstreaming. I tried to stick to time. I may have been quite quick, but the last slide has, I think, or acknowledges our team, and you will also have our handles if you want to get in touch via not just email, which I did not include, but also via Twitter and LinkedIn. I think that's me, but very, very happy to take questions if I rushed.
Fantastic. Thank you very much indeed. Lovely. Well, thank you again. Great discussion once again. And I definitely have my reminder now. So we do need to move on. And the next one is the Express node and Hugh Morris. Hugh.
Thanks. I'm Hugh Morris. I'm a neurologist and geneticist at UCL, speak on behalf of this node. Next slide, please. Our team, the core team involved in this is Tim Rittman, James Rowe in Cambridge, leading on imaging, Michelle Hugh in Oxford, leading on digital biomarkers, Yoav Ben Shlomo in Bristol, leading on epidemiology and cohort development, myself, and a massive shout out to Riona Fumi, who's on the call, who's really been managing the whole project so far. And this is a picture of Riona presenting at a patient meeting at the end of last year, explaining about the study. I would say as well, of course, that our network is very much a partnership with patients. And I think in this area, we've been very fortunate in that we've got very strong links with 2 patient organisations, MSA Trust and the PSP Association, that really have supported our research and support patients and are very keen to help engender networking and development of research in this area. Next slide, please. So as background, these conditions, so this is obviously clearly a disease-focused node rather than a sort of cross-cutting node. Focused on some conditions, PSP, corticobasal syndrome, multiple system atrophy, and early onset Parkinson's plus conditions. These conditions are usually initially diagnosed as Parkinson's disease, but it becomes apparent after they've been diagnosed that they're not Parkinson's disease, but are more malignant conditions that progress more quickly. They are progressive. People lose the ability to walk, speak, and swallow. They're very similar to motor neuron disease, ALS, in the way that they affect people. They progress slightly more slowly, but they are life-limiting conditions. Average age of onset in 50s for MSA and 60s for PSP and corticobasal syndrome. So these are rapidly progressive degenerative conditions. Next slide, please. So really sort of echoing what Bob was saying earlier about the real challenge in trying to deliver trials and treatments for this patient group is sort of underdiagnosis, lack of awareness of these conditions, and a very long delay in diagnosis. So we had a previous network running called the Prospect study. So the average delay from symptom onset to diagnosis was around 3.5 years for these conditions. So unfortunately, what that means for clinical trials is, given that clinical trials usually have disease severity criteria, so people have to be usually independently ambulant when they enter a trial. They have to have a definite diagnosis. There's kind of a squeezing of the window for patients who are eligible to enter trials, and that represents a massive problem for us in getting patients into these trials. Things are developing very quickly in terms of biomarkers that might support early diagnosis of these conditions. So that really is a big focus of what we're trying to do in the Express node, is trying to implement some of these biomarkers through our network to try to enable early diagnosis. So the particular challenge is underdiagnosis, the support that patients get, and of course they get great support from the patient organisations, but if they haven't been diagnosed with the condition that they have, then they're not likely to get the appropriate support and advice. We have a relatively limited knowledge of disease biology, particularly in the early disease stages. So these conditions, of course, haven't really been studied in the early disease stages because people don't get diagnosed till 3 to 4 years after they've started, and diagnostic latency. So our sort of approach to this is the development of this node, the Express node, early assessment, diagnosis and treatment of Parkinson's plus conditions. Next slide, please. So our 4 main goals, first of all, to establish our network across the UK, to establish a platform for early diagnosis, for the development of both biomarkers and therapeutic trials and sort of basic science discovery based on recruitment of early patients. The discovery part of it is based on genetics biomarkers, both imaging biomarkers, but also genetic and fluid biomarkers, and development of new disease models based on that. And of course, I think the ultimate success for this node and for this project will be to establish new clinical trials, hopefully effective clinical trials that will help patients with these conditions and prevent the condition from progressing. And of course, that means collaboration both with basic science, collaborators, academic, and in industry in the development of new trials. So obviously, a lot of what's been said earlier is very relevant to what we're trying to do. Next slide, please. So we had before the Express node was funded, we had a network that was funded by the MSA Trust and the PSP Association called Prospect. That was tapering down actually when Express started because it was coming to the end of the funding cycle. But what we've been able to do is to sort of reinvigorate the study sites. And so far, 17 study sites have agreed to be part of the Express study. And so they're all in progress of being set up as study sites, recruiting patients across the UK with these conditions. And the current study sites are listed here, and there are likely to be more study sites joining in with the project. Next slide, please. This represents the spread of recruitment that we've had previously in the Prospect study. And although the study sites are sort of split across the UK, we've had quite widespread recruitment apart from north of Scotland and Herefordshire for patients from the UK. Within our prior Prospect study, we recruited 1,500 participants over around 8 years. We do a yearly audit of the disposition of all of the patients that we've recruited in this study, and actually the audit's just been done, and about 850 of the patients we've recruited have died, unfortunately. So that kind of gives you a flavour of what we're up against in terms of rapidly progressive condition. Of course, although we've recruited these patients and we've collected biomarkers and trying to study their natural history, very few of these patients now are going to be eligible for clinical trials because of the progression of these conditions. So there's a need to kind of continue to recruit patients in the early disease stages who are going to be eligible for clinical trials. Next slide, please. Another part of the networking activity is integration of the UK Brain Bank network. So currently there are 5 brain banks that are able to study patients who've been recruited to the Express node. Of course, this is a rapidly progressive condition, so unfortunately people do die, and very many patients are happy to agree to donate their brain after they've died to be studied to help people in the future. In terms of biomarkers, really a gold standard in terms of the accuracy of a sort of protein pathology based biomarkers that it correlates with the post-mortem pattern, which is really of course the gold standard for diagnosing these conditions, which overlap and can be confused with Parkinson's disease and other conditions in life. So having a gold standard for diagnosis is extremely important. Also, there's obviously a lot of insights that can be derived in terms of disease biology based on post-mortem studies, particularly when they're integrated with things like genetics and clinical data that have been collected during life. So this is we think a very important part of our study. Next slide. We engage a number of PPIE events, and obviously again we're very fortunate with the PSP Association MSA Trust. So attend a number of study days to promote the project, discuss the project, and we have ongoing links with the 2 main charities to support this study. Next slide. So moving on to the sort of platform part of this, actually establishing how we're going to recruit patients and engage them with research. We spent about 6 months actually redesigning what we're going to do and designing how we're going to take the study forward. Then this year been through ethical approval, or ethical submission, resubmission, and approval. So this was approved actually in August. So we're now ready to start to recruit patients to the Express node via this study, and we're in the process of setting up sites to recruit patients at the moment. Next slide, please. So obviously the components of this include consent, which allows patient recontact and allows sharing of samples and data with collaborators within the Rare Disease Research Network, but also with other collaborators on a sort of global basis who may be able to use data and samples to derive new insights into the condition. A brief clinical assessment, DNA collection, biomarker collection, including CSF, skin biopsies where possible. James Rowe, Tim Rittman leading the MRI part of this, so actually studying MRI features will enable very early diagnosis of these conditions. And Michelle Hughes leading digital biomarkers, so has a number of smartphones that can be deployed to patients where measures can be captured, which will be, we hope, diagnostically useful in defining these conditions early in the disease course. Next slide. In terms of discovery science of this, a lot of this is dependent on external funding, additional grants that can be leveraged from the sort of platform and network we're developing. So Tim Rittman in Cambridge has a couple of proposals on actually getting additional funding for research-based MR imaging, one of which has been successful, another under review. James Rowe has a Parkinson's Plus Centre in Cambridge, which is supporting the Express study, which is enabling recruitment of patients. Ed Jabari here in UCL has a CSF biomarkers proposal that's under review at the moment, which is linked to the Express study and would use samples, if successful, would use samples that have been collected within the Express study. We have the GP2 program, which I'll say a little bit about in a minute, and we have a number of supplementary funding applications underway to try to increase the biobanking, the biosample recruitment within the Express study, which is obviously an expensive part of the study. Next slide, please. So I think indicating the sort of interest in this, we've had a number of requests for letters of support, letters of collaboration for people setting up new assays and new tests. Oops. Okay, yep. So a number of letters of support requested for providing samples to external investigators that are not connected to the Express network. Obviously, we've provided these letters of support and hope we'll be able to share samples to enable research to be carried out by other people. Part of this involves biomarkers, but also PET scanning, looking at tau protein in the brain, and a study potentially linked to industry looking at early diagnosis of MSA. The Express study has been incorporated into the Global Parkinson's Genetics Program, which is funded by Aligning Science Across Parkinson's and the Michael J. Fox Foundation. Effectively, this means that every sample that or every patient that we can recruit within the Express network will have whole genome sequencing that will be funded by the Global Parkinson's Genetics Program. So that's significant added value to our research, and will generate a lot of data that's linked to the patients that we recruit within the study. We are planning for some disease biology meetings now the network or the patient recruitment is up and running during 2025. Next slide, please. As a sort of taster of the sort of stuff that can be done, this is work that Ed Jabari's done. This is looking at seed amplification assay, so looking at a test based on proteins that occur in the CSF that have the ability to seed the aggregation of abnormal proteins. This is potentially a highly sensitive marker for the early diagnosis of these conditions with specific assays for tau and synuclein, and this is work that he's developed based on these assays in CSF that's been collected in patients that we recruited previously in the Prospect study. What we don't know, I mean, of course, a lot of these biomarker studies actually in people with well-established disease, so people have had disease for 3 or 4 years. Of course, the key thing is whether these are going to be sensitive when patients are first referred from their family practitioner to secondary care. So when patients first arrive in clinic, before they start this diagnostic odyssey, can we actually implement biomarkers that can identify people very, very early in the disease course, which of course is when we think they need to be recruited to clinical trials. So that's obviously, and I think, just as an aside, I think that in a way, and the sort of things that we deal with, specialist centres are the worst because we often see people who've been seen 2 or 3 times before and are coming for us for a third or a fourth opinion. And really, we need to change our strategy and to start recruiting people very early in the disease course when they've first been referred in from primary care for assessment. So that's why the network of study sites we're trying to develop across the UK is absolutely crucial to try to identify patients early in the disease course. Next slide. So in terms of industry collaborations, UCL and Oxford are currently running the Horizon study, which is sponsored by ONUS. So this is an antisense oligonucleotide against alpha-synuclein. Really as a kind of, I'm sure proof of principle is the right word, but as a sort of amplification of what I said, it's been extremely hard to recruit patients to this study, despite the fact we have a lot of MSA patients that come both to Oxford and to UCL and to link sites. Because they don't meet eligibility criteria, they're too severely affected by the condition, or they're too old to be eligible for the study. So it's been extremely challenging to recruit people to this study, and that's why I think we need a sort of early diagnosis platform framework, which will enable us to recruit people who will actually be eligible for the trial. So far, obviously, no one who's been recruited through Express has been recruited to this study, but I would anticipate over the next 6 months that patients who are identified through the Express node will be recruited directly to study. There are a number of other studies that are in the pipeline. There's a new study for PSP, Prosper, sponsored by the Ferrer company. So this is an effective anti-tau aggregation drug. Again, they're going to be looking to recruit early stage patients, and I'd anticipate the patients recruited across the Express network will be recruited to this study. There's another study planned for vaccine therapy, GemVax, and there's a study planned for development of a tau tracer for early diagnosis. And for these 3 studies that are kind of in setup, if you like, on the right, I'd anticipate the Express study will feed into these studies, and hopefully this will encourage other industrial partners to come to the UK to study these conditions because of the network and the platform that we're setting up. Next slide. So in terms of how this sort of interacts with all the parts of the rare disease research network. First of all, of course, our study is a bit different in that we're focusing on a rare late onset disease, and we're very much focusing on the diagnostic process. So this is a non-genetic diagnostic process. This doesn't rely on DNA-based diagnosis. This relies on clinical diagnosis, which we're trying to improve through interaction liaison with non-specialist centres, and perhaps this model might be used for other conditions. We have strong links with disease charities, which I think is a major asset for us. Next slide. The challenge is obviously trying to reach out to non-specialist centres. One of the challenges is try to integrate things to clinical practice. So we set up some online self-completion questionnaires for patients and online self-completion questionnaires for doctors who are seeing patients, which are extremely brief and should be able to be done in a matter of minutes after a clinic consultation, to try to minimise the burden for as much as possible to try to recruit patients. This includes online consent and completion of consent online for patients. I think a challenge for us currently is NHS recruitment pathways and waiting lists. So we've talked about trying to recruit people as quickly as possible. And of course, in our meetings with study sites, many of them told us they have 9 to 12 month waiting times for patients to be seen who are referred with suspected Parkinson's, which obviously is a massive barrier to recruiting patients to research for our studies. Collecting biosamples is expensive, so we need to raise more money to try to maximise the benefit of what we can achieve within the network. I think as time goes on, this is what we've seen with previous samples, is especially with CSF, which is limited number of aliquots can be prepared. We have to decide which studies are the highest priority. So we have to really pick and choose between studies and try to prioritize how we use our samples to make sure they're used as effectively as possible. We have minimal knowledge of early stage disease. There is always the interaction between what we do with drug trials and observational studies. And previously in network activities, we've had an agreement between PIs that if patients want to enter a drug study, then they may drop out of the observational study, because for many patients, being involved in a drug study is more important than being involved in observational activity. And some commercial companies ask that people effectively drop out of further follow-up if they enter a sponsored commercial study. So if patients want to enter drug trials, they would take priority over sort of network activities. Next slide. So that's my presentation. I've talked a bit about trying to build up our clinical research network, build up a platform for recruiting patients across hopefully more than 20 study sites ultimately in the UK. talked about what's coming really with discovery science and what we can do to support this, and obviously I think the sort of end measure of success is going to be the number of collaborative projects, number of samples we distribute, the number of trials that we can support, and obviously what we can discover amongst the core investigators in terms of the science of these conditions. Thanks.
Okay, brilliant. Thank you. And we'll move on, if that's okay, to the last of the node presentations, which is REOLUT, which is Bill Newman. Bill.
Thank you, Dave. Thank you, everybody, for taking the time to listen to our work. Next slide, please. So to set the context, our work focuses on disorders that predominantly affect children, that affect the bladder and the ureters, and this has been an under-resourced area. Myself and colleagues have worked in this area for a number of years, and we have made some discoveries around the causes, genetic causes, of some very rare, severe lower urinary tract disorders. But importantly, lots of children are affected by this disorder, this group of disorders, and we hope that some of our work will provide some insights into these more common disorders associated with bedwetting and incontinence and urinary tract infections. And as you can imagine, these broader group of disorders have enormous impacts on children's well-being. Next slide, please. So our major goals are really to really develop and enhance the infrastructure across the country so that we get more people interested in this area of research, and we really engage with the clinical teams so that this becomes a sustainable program of work beyond the NIHR MRC funding. And right at the beginning, we've very much focused on including junior researchers, both clinical and non-clinical, in our team, and that's something that we want to expand upon. One of the things in terms of thinking about the sustainability of our work is that we've put some energy led by Mel Chan, working with Lucy Plum and Joe Clothier to set up a rare lower urinary tract resource within the well-established UK RDR database, which is a well-established kidney database. And we got approval for that in March, and we've already recruited 175 individuals into that database. So our three programs of work focus around new disease gene discovery for these rare disorders, working on some of the mechanistic insights. We know that the bladder is very much controlled, as all organs are, by the nervous system, but also understanding how smooth muscle can be affected and understanding some of the fibrotic processes. And then the third area is on gene therapy. So next slide, please. So this project one is the area where we've been able to make the most progress, but part of that was because this was how the programs of work were designed. We've set up regular meetings, so it's led by Professor Adrian Wolfe in Manchester, myself, and Melanie Chan. Melanie is a nephrologist who's working at Imperial at the London Institute of Medical Sciences, and has just had her fellowship funding extended for another year linked into this work, which is fantastic news. A lot of our energy has been based on looking at setting up new sites of recruitment, working with paediatric urologists and nephrologists around the country, and I'll touch on that in a minute. We've made some interesting new discoveries, identifying variants in genes related to Shinsel-Gideon syndrome and to DSTYK. We're doing some work with the EpiGenRare with Sid and his team, starting to design a project looking at methylation profiles in bladder extrophy, and Mel's undertaken some analysis of a condition called posterior urethral valves, where she's looked at data from over 200 children and compared those to over 10,000 controls. So we're making progress. We've got some exome data and genome data that we're regularly analysing and linking in with international colleagues in this space. Next slide, please. So Project 2, this is led by Professor David Long at Great Ormond Street and Professor Mena Clapworthy at Cambridge, and has just started fairly recently, but they've made some excellent progress and are hoping to analyse their first tranche of data later this month. So they're looking at expression profiles of different genes in different cell types relevant to the bladder. So for example, the epithelium, and they're doing that in normal tissue, in bladder exstrophy tissue, and in tissue taken at surgery from patients with posterior urethral valves. As you can imagine, there's some technical challenges around this, so organising the protocols, making sure that you can get the right numbers of nuclei from the cells, particularly from fibrotic tissue, is really challenging, and so they've made some really good progress just on that initial part of the work. Next slide, please. And the third major research program is really focusing on new types of treatment. So this is led by Neil Roberts, who's a lecturer in Manchester, working closely with David Long and Adrian Wolfe. Neil has taken a discovery of a gene that we identified associated with the condition called urofacial syndrome a number of years ago, taken that through a number of mechanistic studies, and then to generating a mouse model, which he has been able to correct using adenovirus therapy, which is really exciting. And what he's doing now is really looking at understanding how can you transfer that information into humans. So both at a technical level, looking at what are the best vectors, what's the best clinical approach to that. But he's doing a lot of work, and this is linked into a program of work in Manchester and with our patient support group, at trying to understand, is gene therapy something that parents want for their children in the context of bladder disorders? There are no gene therapies for bladder disorders, and so the patient voice is really important in this particular space. Next slide, please. So we've had some significant success with funding, particularly for Neil, who's got a large project grant with Adrian from the MRC this year, and working with a paediatric neurologist at Great Ormond Street, Nav Johal, who's got one of the MRC CARP awards, which is really fantastic. And just to flag around the Life Arc program of work, David's work is very much linked into that. He's got a sub-program within the kidney centre, which has been awarded over £350,000. So there is some further funding that has come to light as we've been putting together our annual report, which is really positive. We've had a number of publications, but I just wanted to highlight the work from Neil, led by Philippa Lopez, around their gene therapy work, which is a completely new approach in bladder disorders. Philippa's just had a baby, which is wonderful news for our node, but it means that she's been off, and when she comes back from maternity leave, that Project 3 will really progress from there. We had an excellent networking event back in June, hosted by David and the team at Great Ormond Street, where we heard the perspective of the team from ERIC, who are our charity partner, but a number of young investigators all presented their projects, and this was something that we saw work very nicely from John Sayers' Silaren group, which we attended their meeting in Newcastle, and we were really impressed by how they got so many young investigators standing up and talking about their work. Next slide, please. So a lot of our work and energies have been invested into spreading the word a little bit about our new initiative, particularly talking at a number of European meetings. We had a dedicated seminar on lower urinary tract disorders at the European Society of Human Genetics, where Mel and Neil both spoke. I was in Belfast a couple of weeks ago talking to all the British paediatric urologists, encouraging them to become new partners with us to set up sites and to help with our recruitment. Next slide, please. And so one of the things that we've done in Project One is to set up a student placement for an overseas student, PhD student, who's going to come and work with us. We've had some very nice intra-node working, where we had a new candidate for bladder extrophy. We think a gene that hasn't been associated with this disorder previously looks very encouraging from a genetic standpoint, and working with David Long and his team to really understand the mechanistic elements of that to give us further confidence around that. As I said, Project 2 has just started, but it will really, the next steps will be driven by the data that's going to be analysed this month. And Neil's working on his gene therapy programme, and that will really kick off in earnest when Philippa is back. So one of the things that we've got next month is a talk in Birmingham with our charity partners. We'll be talking with them about how we can engage and what are the major questions for them that they would hope that we would address. And Adrian is speaking in Valencia to the European urologists. Like the EpiGenRare and the ELSI node, we are having our node day the day after the whole node conference in Manchester, and so we're very keen to see people there and have an opportunity to see where our work overlaps. Next slide, please. The main challenge, I think, so far has really been the logistical challenges in setting up the new sites. The paperwork around the ethical approvals and site-specific approvals has been frustrating. That's delayed our getting some of the recruitment into the study up and running. And of course, we're working with incredibly busy clinicians. Pediatric urologists have a very heavy workload, and finding time to commit to our program of work is hard for them. And so trying to make things as easy as possible is going to be really important. And I think that hopefully I've already touched on what we think the biggest impacts are. If we really are successful, we will have developed and delivered the first gene therapy for a bladder disorder. We will have provided really significant insights into how rare diseases tell us about much more common diseases, and we'll have understood some of the mechanistic underpinnings of some of these disorders. I think that's all my slides, except for just to say thank you to all the people who have helped, and particularly to Henry and Gabby, who are our project managers here in Manchester. Happy to take any questions.
Bill, thank you. That was great. On 2 o'clock. So my colleagues who've been messaging me all the way through saying, hurry up, we're not going to finish on time. It's time. So thanks, everybody. Brilliant discussion. I really, really enjoyed it. And we've got so much to follow up on. So look forward to seeing you all again next time. So thank you.